Now moving on to Kraken, which is an ultra-fast metagenomic sequence classification method given by Wood and Salzberg in 2014. So the core concept is basically Kraken is designed to classify every read in metagenomic datasets. It uses exact k-mer matching, default k is equal to 31 NT, against a pre-computed database of reference genomes. Each k-mer is associated with the lowest common ancestor LCA of all genomes containing that k-mer, right? For a given read, all its k-mers are mapped, LCA is retrieved, classification tree scored, and we take the best taxonomic path out. Kraken databases, they require pre-built or user-specified databases. Default databases contain bacterial, archaeal, viral, and human genomes. Users can build their own from repositories like GTDB, which is Genome Taxonomy Database, high-quality curated microbial genomes. Kraken 2 and microbial databases, for example, Loman Lab mock communities for benchmarking. Pre-building is expensive, hundreds of gigabytes to terabytes of RAM required. GB to TB of RAM is required, so it is expensive depending on the database size. So step-by-step classification algorithm. Step 1 is to take the input, a read that is a length L, for example, 100 base pairs Illumina read. Then extract all overlapping 31 mers, L minus k plus 1 k mers. 100 base pairs read basically takes 70 k mers, right? If k is equal to 31, if you're considering k is equal to 31, according to this formula, L minus k plus 1. LC lookup for each k-mer, Kraken queries its database hash table. Each k-mer points to the LCA node of all genomes where this k-mer occurs. Example, k-mer appears in E. coli and Salmonella enterica. So LCA is the family Enterobacteriaceae, right? Then path collection, collect all LCA nodes from k-mers, expand each to a root to leaf path RTL in the taxonomic tree. Scoring. Now for each RTL path, add weights for each k-mer supporting a taxon along that path. Scoring reflects how many k-mers from that read support the classification. Example, path to E. coli is 50 k-mers, path to Salmonella is 20 k-mers, and path to Enterobacteriaceae is basically the sum of these two. That is the root to path sum up till the LCA, which is this. LCA is the least common ancestor, lowest common ancestor, which is 70 k-mers when we sum these two, right? Classification, the maximal scoring RTL path, root to leaf path is chosen. Read if assigned to the deepest node supported by sufficient k-mer evidence. If all k-mers are too diverse, classification stops at a higher taxonomic rank, for example, phylum. Now, trade-offs in this design. So advantages, firstly, we'll consider, first, it is ultra fast. Exact k-mer lookups are hash-based, O(1) operations, classifies millions of reads in minutes. It classifies all reads. Every read is assigned, even if only to a high-level taxon. Broad coverage, works across bacteria, archaea, viruses, eukaryotes, human, etc. Customizability. Users can build their own databases with specific genomes, for example, using the Genome Taxonomy Distribution database, GTDB, right? Example, clinical pathogens in environmental species. Scalability can extend to new environments, marine, soil, gut, wastewater. Disadvantage is memory intensive. Database requires hundreds of gigabytes to terabytes of RAM. Kraken to reduce this, but trade-off is in sensitivity. Computationally expensive build time. Building large databases takes days on high memory clusters. False positives. Reads with common k-mers shared across genomes can be assigned to incorrect taxa. Sensitivity versus specificity. Very sensitive, classifies every single thing, but noisy if database incomplete or contains misannotations. Limited novelty detection. Cannot handle species absent in the database. Falls back to higher level LCA. Now Kraken versus alternatives. So Kraken takes the exact k-mers, ultra fast and RAM heavy. Kraken 2, memory efficient probabilistic database compression, slight sensitivity loss. Metaflan, which is metagenomic phylogenetic analysis, uses marker genes, plate specific marker genes, slower but highly accurate and less memory intensive. Blast, right, alignment based, accurate but orders of magnitude slower, and centrifuge similar to Kraken but optimized for low memory environments. Now, practical implications best suited for large shotgun data sets, microbiomes, clinical metagenomes, environments where classification speed greater than absolute precision, scenarios with access to high performance clusters or cloud resources. Not suited for resource limited environments, low RAM, studies needing absolute precision for rare or novel taxa. Right, so that's it.